Bismillahirrahmanirrahim. The topic of our discussion is biological species concept. We have previously discussed its various points and its importance and its applications. However, there are still certain difficulties in the application of the biological species concept, which will be discussed today. There are certain difficulties which sometimes arise in certain situations in the application of the biological species concept to the natural taxa, which does not mean that the concept is invalid, because in certain situations the two species concepts previously discussed, biological species concept and typological species concept, are basically used for facilitating the process of identification. The similar situation is faced in the application of various other accepted concepts. For example, if we consider that the importance of a tree, that cannot be rejected due to the existence of certain vines and herbs. The most serious difficulties in the application of the biological species concept are those caused by insufficient information, uniparental reproduction, and evolutionary intermediacy. These 3 points will be discussed in the future slides, in the upcoming slides, in detail. Insufficient information. Individual variation in all its forms often raises doubt about whether a certain morphotype is a separate species or only a phenon within a variable population. As we have previously discussed, that phenon refers to the phenotype of an individual within a population. So there might be different phenom of a species within the same population. For example, if you consider ants, we do find that in a colony there are different phenotypical variation between the organisms of the same colony or within the ants of the same colony. Similarly, if you consider a frog, during its life history, it transforms from one morph to another morph. That is, from egg to tadpole, tadpole to froglet, and froglet to an adult frog. So there are various morphological variations within the same population, and we need to have sufficient information about the life cycle of an individual, and also the population analysis should be very complete. Sexual dimorphism, age differences, polymorphism, and other types of variations can be unmasked only if we study a complete life history of an individual and we go into the population analysis of that particular animal. The neontologist, a person who normally works with the preserved material and in the museums, faces similar problems that a paleontologist confront, and they generally assign morphotypes to species. Uniparental reproduction. In certain organisms, there is no sexual reproduction, and systems of reproduction in those organisms are not based on the principle of recombination of the genetic material between the parental organisms during the formation of a new individual. There are certain examples, that is self-fertilizing, hermaphroditism, automixis, parthenogenesis, gynogenesis, vegetative reproduction, that is budding and fission. These are mostly frequent in lower invertebrates and even in certain lower vertebrates up to reptiles. So insufficient information and uniparental reproduction are the limitations of the application of biological species concept. Whenever there is some kind of insufficient information of an organism, we cannot apply biological species concept because we do not know the wildlife of that particular animal. Similarly, if there is uniparental reproduction, we cannot apply biological species concept because there is no recombination of the genetic material between two different organisms. What is the definition of a population? If you are considering it in evolutionary biology, a population is defined as an interbreeding group. Whenever we are considering an asexual biological population, this becomes a contradiction to its true and original definition. So the word population in general terms has also other uses, and for that asexual members can be referred using this term. Interbreeding is the ultimate test of conspecificity. Basically, when we say conspecific, we mean that the two members are belonging to the same species. So whenever we have to test whether the two members or the two morphotypes are belonging to the same species, we have to check whether they can interbreed and produce fertile offsprings. And this criterion can only be applied in the sexually reproducing organisms. So whenever we are considering the uniparentally reproducing organisms, it becomes difficult to assign a categorical rank to that particular taxa. So what should a taxonomist do to treat clones, pure lines, biotypes, or certain strains or stocks of such organisms which are uniparentally reproducing? So such uniparental lineages are designated as agamospecies, binomes, or paraspecies. Whatever designation one chooses, one must be aware that such entities or such groups are not subdivisions of the biological species, but they are quite different. And it was rightly questioned by certain scientists that the propriety of applying the word species to groups of asexual clones, and according to Meyer, they were generally being agreed by most of the taxonomists. In some groups of animals, particularly aphids, gall wasps, Daphnia, rotifers, and digenetic trematodes, a regular alternation of generation between sexual and parthenogenetic generations occur. In such cases, neither kind of generation qualifies for separate species status, as nomenclatural recognition is not given to temporary clones. However, the parthenogenetic generations sometimes fail to return to sexuality, particularly in aphids, and become permanent. When they differ from the sexual races by host plant preference or even by color genes, the suggestion that these parthenogenetic taxa should be called species deserves serious consideration. Once it becomes confirmed that the different morphs are not the alternative generations of the same organisms, the morphotypes are completely studied and understood, and once we find a permanent uniparental reproducing line, then we assign a species status on the basis of degree of morphological difference to delimit that particular species. Delimit means that we put a boundary around a species on the basis of certain morphological markers. The morphological discontinuities or the morphological variations among two different species, which we are going to consider as different species, must be well defined and apparently produced by natural selection among the various mutants that occur in the asexual clone. As it becomes confirmed that different morphs are not polymorphic forms or alternate generations of the same animal, we begin to consider the morphological discontinuities as markers for identification of the species. We recognize species by comparison of the characters or comparison of the morphological discontinuities, their ecological niche, and the evolutionary roles they play in their ecological niche. For example, a form that is as distinct as a good species reproduces strictly parthenogenetically, and no biparental species is known from which it has branched off, and we give a nomenclatural recognition because it is justified in such cases. Whenever several reproductively isolated chromosome types, for example, occur in a species, as we see in different crustaceans, it may be convenient to distinguish them nomenclaturally, although they are conventionally referred as races. It is more logical to designate reproductively isolated chromosomal populations as microspecies. There are nearly 1,000 animal species in which the males are either absent or non-functional in the reproductive process. One phenomenon is the theletoky or Theletochus species, and the other is kleptospecies. It depends on the meiotic mechanisms that what kind of offsprings are generated. Either they are homozygous or they are heterozygous. Homozygous are rare as it is restricted to few insects. Along with these, sometimes sibling species members are also present in nature that are bisexually reproducing. Sibling species are those species which are different species, however, these are morphologically similar. As we have previously discussed, that asexual organisms are identified on the three criteria, that is, morphological discontinuity, their ecological niche, their evolutionary roles, but we cannot consider the biological or reproductive isolation. Considering the heterozygosity promoting theletoky resulting into the production of heterozygous offsprings are the result of certain speciation by switching to the parthenogenesis in an individual who has originated from interspecific hybrid. Kleptospecies. The male chromosomes are lost during meiosis and fertilization by males of one parental species is needed to induce the development of the egg, even though the males do not contribute to the genotype of the developing egg or developing zygote. We have some examples in frogs. Hybridization between two distant species of animals apparently always result either in total sterility or in abandonment of the sexual reproduction. We have already gone through it in the biological species concept, in which we say that if two distant species try to interbreed and they become successful, then the future offspring would be sterile. Or we can we can see that there might be an abandonment of the sexual reproduction in the future generations as well. In groups with cyclical parthenogenesis, the sexual generation may be permanently lost in some species. And if there is a switch in a host species or a host species is changed, for example, in aphids we can see, then a doubt in the species status is raised, and we say that possibly the species must be rechecked or reconfirmed. Hermaphrodites in most cases reproduce sexually, that is, fertilization of eggs is effected by the spermatozoa of a different individual. However, in certain cases, there is a practice of complete automixis, which is self-fertilization. This results in increasing the homozygosity found in several species of slugs. The next limitation of the biological species concept is evolutionary intermediacy. The species are defined on the basis of the reproductive isolation in non-dimensional situation, that is, space and time. So we can say that under biological species concept, the allochronic and allometric species cannot be considered, as their reproductive isolation cannot be checked. As we add these two dimensions, then the inclusion of incipient species is also possible. Incipient species are those that are in the process of becoming new species and have acquired some but not all characters of a new species. So the question is that at what point a diverging population is called a species? It becomes difficult to decide when the morphological distinctness is not supported by the reproductive isolation. The difficulties a taxonomist faces due to evolutionary intermediacy will be discussed in the upcoming slides. So the difficulties the taxonomists face due to evolutionary intermediacy are: number one, acquisition of reproductive isolation without equivalent morphological change. So at this point of time, there are certain sibling species which are morphologically very similar, however they are reproductively isolated. So reproductively isolated species without or with very slight morphological difference are called sibling species, and that is the point where taxonomist faces difficulty in the application of the biological species concept. Number two, acquisition of strong morphological difference without reproductive isolation. Number one, morphologically very different populations interbreed wherever they come in contact. Genera in which the isolating mechanism between two species break down occasionally. So this is another problem which is generally faced by a taxonomist during the application of the biological species concept. So no generalized solution is possible in cases where morphological divergence and acquisition of reproductive isolation do not coincide. This is an example of irregular distribution of population of a land snail in eastern Cuba, where the numbers are showing the races and the species, and where the two populations come in contact and they hybridize. That has been represented by age regardless of their difference, and in certain cases they are being prevented by a barrier which is represented by B, and IN is the isolated population which are being shown in the same diagram. You can see it at different places. The occasional breakdown of isolating mechanisms leads to hybridization. Reproductive isolation may break down occasionally, even between two good species. Usually this leads only to the production of occasional hybrids, which, being either sterile or of lower viability, do not cause any taxonomic difficulty because they do not survive or they do not continue. However, sometimes or very rarely there is a complete local breakdown of isolation that results in the production of an extensive hybrid swarm and more or less complete introgression. Introgression is the transfer of the genetic material between species following hybridization, and then those hybrids backcross to their parental species, resulting into hybrid swarms or parthenogenetic species formed through hybridization. In hybrid swarms, we have certain species where the reproductive isolation is maintained over most of the area of the sympatry, which may break down locally, resulting in the production of localized hybrid swarms. Whereas in the parthenogenetic species formed through hybridization, results in the production of an individual with one chromosome set from parental species A and one from parental species B, and leads to the doubling of the chromosome set called allotetraploidy. Hybrid swarms. These hybrid swarms are produced as a result of the breakdown of reproductive isolation in local sympatric populations. These new hybrids are not given the species status, and species status of their parent is upheld or considered. The species status will only be given to a hybrid if there is a production of a new species. Parthenogenetic species formed through hybridization. Production of individual as a result of fusion of a complete set of chromosomes from parental species A and one complete set of chromosomes from parental species B results in the doubling of the chromosomes called allotetraploidy. Allotetraploidy in animals lead to the parthenogenetic mode of life. These are morphologically distinct and are generally recognized as valid species. Semi-species and allospecies. There are certain geographic isolates that cannot be considered as species. They are referred as subspecies and are put under species to which they resemble most. On the other hand, some of them may reach the species level and are called superspecies. These populations are referred as allospecies. The cases like these are solved on the basis of degree of evolutionary intermediacy. Taxa that are intermediate between species and subspecies are referred as semi-species. Species concept facilitates the delimitation of species taxon, or it helps to put a defining boundary around the taxa of a particular species category. Monotypic species are not variable, so can easily be diagnosed and identified. The problem is generally faced by the taxonomist during taxonomic characterization while working on the sibling species, number one, and number two, on the allopatric or allochronic species. This problem is solved through by working through this problem is solved when we are using biological species concept, as it facilitates the process of identification, classification, or the species delimitation. This can also be facilitated by inference or logical judgment, which can only be done through observation, and especially by a person who is working in the field of wildlife. This is most reliable than any other person can even expect. In an analysis of 607 species of North American birds, Mayer and Short found that biological species concept was unable to clarify only a single case. It permitted the settling of all problems and controversies with respect to polymorphism and sibling species. 45 cases of hybridization among species taxa was solved using biological species concept. 27 cases as intraspecific and 18 cases as interspecific hybridization. Among 607 North American species, 247 were polytypic, but only 46 of them were contain populations that some authors consider full species and others consider subspecies. They were probably the peripheral isolates. Since populations are isolated, the biological species concept cannot be applied directly on them. So it is a sympatric situation that is of particular importance for the ecologist and the student of behavior. There is no other species definition which can provide a foolproof system for the correct assignment of the isolated populations or other cases of the evolutionary intermediacy. So we work with all the species concepts, that is biological, that is typological, and now people have also started working with certain genetic markers that are too species specific, for example barcoding. But these species specific markers have their own limitations. So we have to work with all the concepts, and we have to work with all these concepts, and we use them for the facilitation of the identification, and we use them as our confirmatory tests. So if you do have any questions, you can ask it on the same forum. Thank you so much. This is all about the species category.